Oh yeah, okay. So first introduce my colleagues Henning Rudolph and Carmen Waengler from the Universitätsmedizin Hospital in Mannheim and Constantin Mamat from Zentrum Dresden-Rossendorf, because I had the impression we already worked with lead-212 and lead-203, but we needed more for doing preclinical research, and I thought I will get it and I could distribute the rest I don't need to them, and it worked out. So first, here a small introduction of the isotope I talk about. Okay, let's see. It's here. Yes, we start with radium-224 and end up at lead-212. And lead-212 actually is not the alpha isotope because it decays like with a beta-minus decay with 212, and then an alpha decay happens either 36% that way or 64% that way, and then we have one alpha at the end. So next slide. Where we start, the clinical background of the matched pair. Lead-203 is a SPECT nuclide with which we can measure and do a calculation of the dosimetry. And with lead-212, we can do afterwards treatment. Lead-212 has only one alpha decay, as I mentioned, and you need doses between 200 and 250 megabecquerel per patient. Because it only has one alpha. And for actinium, for example, we have 4 alpha decays, 3 in the cell, and there we use only 5 to 10 megabecquerel for treatment. So the first treatment of lead-212 was just to Mamat in patients 2014, and then we had dotan-mutate lead-212 from Dalpasan. You know already that they found out the dose of 2.5 megabecquerel per kilogram that worked very well, and it was obtained so for neuroendocrine tumors, and PSMA are in the preclinical state for that time. So what we did, how far are we in our clinic? We already have a cooperation with another company called Perspective Therapeutics, maybe you know them, and they have the BMT alternate, or it's also a PSC PAC2 top derivative, with which we did first lead-203 dosimetry, as calculated a dose of at least 90 megabecquerel of 212 lead ligand, and we treated 8 patients, and yes, and all of them developed a stable disease with that low amount of activity, with very low side effects. And 2 of the patients now, after 1 year, get their second cycle. Now let's switch to the results from Mannheim. Their aim was to develop an automated method on a Gaia Luna synthesis module from Universität, and they actually did it. One thing I have to say, if you work with such a generator, you will have a very high contamination problem with the radium-220, and that is why I recommend every one of you to work with lead-203, use an automated method within a few months. Actually, they did, that is what I show you, they did derivatization of a PAPI 2286 derivative, where they changed the DOTA chelators to a DO3AS chelator and labeled it stably with lead-203, as you can see here, and also with lead-212, where you can see that the DOTAs are forming around the time, and they did also cell uptake studies and found out that their affinity and cell uptake was very high, and the affinity was 36 nanomolar. They had a successful labeling of those 3 AM peptides up to now. Now, the main goal of the PRISMAP project was that we found out that the macropar is not only good for actinium, but it can also stably chelate the lead-203 and lead-212, and it has the benefit that you can label it on 2 sides with, for example, 2 PSMA derivatives, and maybe you think that 2 PSMA could have a higher uptake in the cell, and you see it like here. This is one moiety with 2 PSMA, and this is the other moiety with only 1 PSMA. You see a high uptake in cells. And what we also found out that maybe there could be a rechelation in patient solution because we have the room temperature and 20 and 5 minutes labeling conditions. Yes. So what they found out, yes, this is macropar, and macropar has a very high stability for lead-212, and that is why we choose also to label macropar with lead. So here, the first thing for the free radionuclides we found in patient solutions, for example, with actinium or lead, is that we found the daughter nuclides bismuth 213 or bismuth 212. And current situation is that we add DTPA to the patient solution to chelate the free daughter nuclides from the ligand, that the kidneys can eliminate it faster, because the free bismuth would stick to the kidney, and then you have a higher dose. So what I tried was to do a higher amount of precursor using, for example, here 50 microgram of macro PSMA, 50 microgram of the normal PSC PSMA, and as competitor the 50 microgram of the Otaga PSMA INT, and doing the labeling conditions. You see here room temperature, a little bit higher temperature for PSC, and high temperature 95 degrees for Otaga. Overall, we have nice results for those two here. I have to say, maybe 20 beats and 30 minutes is way too long. We have it already after 5 minutes. And what I wanted to look now is, when I use such a high amount of precursor, do I see rechelation after purification in the patient solution? So what I did was doing for the macro PSMA and lead-212. After 1 hour, I see such a high purity. After 21 hours in reaction solution, I see no free bismuth 212 at all. Okay, 2%, just for seed, and a little bit more in the diluted reaction. But after 44 hours, the amount of non-bounded or non-chelated precursor is consumed, and then you see free bismuth happening. This didn't work for PSC PSMA, for example. PSC is chelator from the BMT alpha-net, from the perspective thing. Macropar was even better, and for Dotaga, it was the same. We always have free bismuth right after the synthesis. So take home for the rechelation in patient solution for targeted alpha therapy is that rechelation can only be observed for actinium and lead-212 with macropar, and the rechelation is more effective in reaction solution than in patient solution. You could think about a company produces the macropar derivative, doesn't matter which kind of, and sends it in a reaction solution, not in a diluted patient solution, for example. And then in the clinic, one have to distribute it to different patient doses, maybe. And the rechelation lasts longer for actinium than for lead-212. Now the thing for the Helmholtz and Dresden, there we had the macropar with different derivatives. So one with a mono PSMA, one with a D PSMA. Then we thought maybe we had the albumine binder here, and then we also had a compound with 2 albumine binders and 2 PSMA derivatives at the site. The reaction was always at 20 degrees for 50 minutes. Very good, very high molar activity. I mean, we use here just 2 to 4 microgram of precursor, and right now in the clinic we use this up to 200 microgram of PSMA INT, for example. So rechelation is possible. QC is also okay. Use this one, and now I show you some in vivo assays from actinium, because for the lead we did not already have it in presentation form, but what you can see here is that we have tumor uptake for the mono PSMA, which is comparable also with the D-PSMA here, and all this results here are comparable with the PSMA 617. But when we go to the albumin binder, this is the mono albumin binder, we have a much higher tumor uptake, which what you can see here, already beginning a small higher accumulation of organs at risk. And for the D-albumin PSMA binder, we have still a rising uptake, much higher than in the kidneys, for example, but as you can see, higher background in organs at risk. Therefore, we decided to look at patients, for example, for this moiety here and for the D-PSMA for the first. Idea on the fly was we have iodine as the albumin binder, so why not just changing the iodine to a 123 iodine and do imaging with that? We compared it, and in mice it looked very good. We have here a high tumor signal for the moiety with lanthanum in it, so stable lanthanum, just to say that this is labeled, and also without lanthanum, you know. So we already also did not see a much high deionization for this compound, and therefore we just said, okay, let's try this with the first patients. 2 patients we had. Here's gallium signal, gallium PSMA with high tumor accumulation, and you see just very few uptake, more or less diffuse background. And the problem was that the iodine or the whole albumin binder got away from the molecule, and therefore we had a retaining activity in the blood pool and not a higher accumulation in the tumor. That is why this compound, the albumin binder, was cut out of the project, and we switched further to the mono PSMA and the D-PSMA derivatives. As you can see here, the monomer and the dimer both have a high uptake after 2 days, and after 7 days, no background, but still activity within the tumor site. And here, the comparison with the albumin binder, as you can see here, we had also a very high background in the blood pool already also after 2 days within the mice, and the mice have a much higher pharmacokinetic and faster elimination from as compared to human. So, but nevertheless, we took this compound and tested this with lead-212 for survival of mice. We performed 200, 400, and 800 kilobecquerel per mice. And as you can see here, this one is the PSMA 617, and those both are the monomer and the dimer, and actually the violet line is the albumin binder. So it looks very good in a mice. Yeah. Now I want to show you the generator evolution in the nuclear medicine. So first, it was the idea just to have the radium-224 attached to the glass wool, and then the radon can go to the glass vial, and when the radon decays to lead, it goes to the sides of the glass, and then you just can rinse the glass bottle and get out your lead-212. But this was really a mess, because in the moment you open the bottle, you get free radon everywhere in the fume hood. So was not a good idea. At the end, we used the PRISMAP piece, the PRISMAP lead-212 from that state on in liquid form with the lead resin, and this worked very well. And other companies like the Perspective already are sending us generators which are produced in the US. So this is from Europe. Yes, and Here I proceeded because we already produced the VMT AlphaNet in our clinic. I wanted to have a GMP-conform method for radiolabeling of peptides, not only of PSMA mutate, but also of others. And therefore, I have the automated elution of the generator here, and this is the modular Easy Lab from Eggert and Ziegler, together with the C18 cartridge purification, and we have here the normal activity curve within the process, and after the synthesis, very good radiolabeling. This here is, I must say, this is already free thallium, which you can see after 1 hour, this thallium signal and also bismuth goes higher, and after 4 hours, it's here. So, and at all, we use this TLC in our clinic to check for the quality. And here I have to say that we use the AR2000 TLC scanner, because we can switch between beta minus detection and alpha detection. And first I thought, because I have an alpha emitter, like 212, no, it is beta minus. The alpha detection for that shows us already actually the bismuth, and we see much higher colloidal bismuth in our solution as compared to colloidal lead-212. So I recommend to use the beta minus detection to do the quality control for your product. And that is when we proceeded to the first human application of macro-RD PSMA, also in 2 patients with lead-203. At this time, it came from Abonams in Nantes. And here's the comparison with the gallium PSMA patient. And what you can see is that we have here a high accumulation in the bone, which we can also see here. But maybe here also a small metastasis lesion, which is also here very high accumulated. But the rest of the lesions we cannot see. So we were wondering, is maybe the resolution of the lead-212, lead-203 too less, or is there another problem? And what we found out is that we also here find a longer blood pool circulation of the dPSMA, because the substance is more lipophilic than normal mono-PSMA derivative. And the lead compound has charge less than, for example, the actinium compound. Actinium is 3+ and lead has 2+. And maybe this is also an effect that we have another biodistribution as compared to the animal. One short excursion for this is that if you use the same substance like here, ED-dotatate, and switch the radionuclide with different charges, for example, 3+, 2+, 3+, 3+, you see always a different accumulation just by switching the radionuclide. And that is why we think we have the same problem here when we switch from actinium to lead. Therefore, the take-home for the preclinical results is that macro-RD PSMA, which have a higher uptake than a mono-PSMA, and mono-albumin PSMA, which have a very strong uptake, are promising, or were promising, very promising in animal studies. But in patients, we saw a different biodistribution. For example, a higher blood pool. And macro-mono PSMA, which have at least a higher molar activity, might be the last candidate for using, for labeling it and testing it in patients with lead-203 and lead-212. And after this turns to be good, we might switch back to macro-RD PSMA, maybe with lanthanum 133, because it has the same oxidation state like actinium-225. Yes. Therefore, I want to thank you, and we now have 5 minutes left, so you can ask me very much questions. Thank you very much.